Cellular respiration. Cellular respiration is a biochemical process that occurs within the cell of living organisms to convert chemical energy from food, primarily glucose, into stable energy in the form of ATP or adenosine triphosphate. This process is essential because ATP serves as the primary energy currency of the cell, fueling various biological activities such as movement, cell division, and molecular synthesis. Cellular respiration is a complex process involving a series of enzyme-catalyzed chemical reactions that take place primarily within the mitochondria, the organelles often referred to as the powerhouses of the cell. There are three main types of cellular respiration: aerobic respiration, anaerobic respiration, and fermentation. Aerobic respiration is the most efficient type, as it uses oxygen as the final electron acceptor and produces the most ATP. Anaerobic respiration, in contrast, occurs in the absence of oxygen and uses alternative electron acceptors, resulting in the lower ATP production. Fermentation is a special form of anaerobic respiration that uses organic molecules, such as pyruvate, as the electron acceptor. Examples include alcohol fermentation by yeast and lactic acid fermentation by muscle cells. Aerobic respiration consists of four major stages: glycolysis, oxidative decarboxylation, formation of acetyl-CoA, the Krebs cycle, citric acid cycle, and the electron transport chain. Glycolysis occurs in the cytoplasm, where one glucose molecule is broken down into two molecules of pyruvate, yielding 2 ATP and 2 NADH. The pyruvate molecule that enter the mitochondria and undergo oxidative decarboxylation to form acetyl-CoA, generating NADH and releasing carbon dioxide. Acetyl-CoA enters the Krebs cycle in the mitochondrial matrix, producing 2 ATP, 6 NADH, 2 FADH2, and 4 CO2 molecules. The final stage, the electron transport chains, takes place on the inner mitochondria membrane, where electrons from NADH and FADH2 are passed through a series of proteins and ultimately combined with oxygen to form water. This process produces about 34 ATP, making the total ATP yield for one glucose molecule approximately 36 to 30 ATP in aerobic respiration. In the absence of oxygen, cells undergo fermentation. In lactic acid fermentation, pyruvate is converted into lactic acid, a process that occurs in muscle cells during intense exercise. In alcohol fermentation carried out by yeast, pyruvate is converted into ethanol and carbon dioxide. Both types of fermentation yield only 2 ATP per glucose molecule, significantly less than aerobic respiration. The key differences between aerobic and anaerobic respiration lie in oxygen usage, energy efficiency, and products. Aerobic respiration requires oxygen, generate more ATP, and produce water and carbon dioxide. Anaerobic respiration does not require oxygen. yield less ATP and result in byproducts such as lactic acid or ethanol. The mitochondria play a central role in cellular respiration, hosting the Krebs cycle and the electron transport chain. Glycolysis, however, occurs in the cytoplasm. Several factors influence the rate of efficiency of cellular respiration, including oxygen availability and presence of fuel molecules like glucose, temperature, pH, cellular activity, and mitochondrial health. Additionally, hormone and genetic regulation also play a role in how cells manage energy production.